Over 2 billion years ago, a type of photosynthetic bacterium evolved, a cyanobacterium, which produced oxygen as a waste as it performed photosynthesis. About 1 billion years ago, a new type of green photosynthetic cell evolved. It was a eukaryotic cell that had cyanobacteria living inside it. This was a second endosymbiotic event, with the first endosymbiotic event involving the incorporation of the proteobacterium, which would become the ancestor of the mitochondria. This was an endosymbiotic event that occurred in an amoeba, which absorbed a cyanobacterium. The cyanobacterium became the ancestor of the chloroplast. But this then would pave the way for a new type of endosymbiosis, where a large cell could ingest a photosynthetic eukaryote, like a red alga or a green alga, which itself then had the descendants of cyanobacteria living inside it. So the ingestion of a bacterium is a primary endosymbiosis. The ingestion of a eukaryotic cell, which has endosymbionts inside it, so that this eukaryote becomes a new endosymbiont. This is what is known as secondary endosymbiosis. For example, there is a group of protists known as euglenozoans. Some are green and photosynthetic, while most are not. It seems as if within this lineage a secondary endosymbiosis occurred, where a green alga was taken inside a euglenozoan. Not the first, because there are many lineages, such as those which cause African sleeping sickness and Chagas disease, which do not have this endosymbiont. So only some of the euglenozoans are green and capable of photosynthesis, those which descended from this endosymbiotic event. But it was a secondary endosymbiosis because the endosymbiont was a eukaryotic green alga. Instances of secondary endosymbiosis would be different from those of primary, because the endosymbionts could have more than two membranes around them, such as 3 or 4. There could be remnants of the nucleus of the original endosymbiont eukaryotic cell, and also the genes which get transferred to the nucleus of the new host would bear a unique signature. Protists such as Cynora, the diatoms, and brown algae are classified together as heteroconts because of a number of shared features, like their flagella, but also because of their photosynthetic pigments, chlorophylls A, C, and fucoxanthin. This fucoxanthin helps to give many of them a brown pigment. Once again, this would be a secondary endosymbiosis because the ancestral heterocont ingested a eubacterial cell, a red alga, which was itself possessing endosymbionts in the form of both mitochondria and the cyanobacterium, which had produced its chloroplast. In the same way, dinoflagellates are a group of protists which can possess photosynthetic plastids which do not seem to be the result of a primary endosymbiosis from the absorption of a cyanobacterium, but rather a secondary endosymbiosis from absorbing a cell which was an ancestral red alga or close to the split point of the red and green algae. These dinoflagellates may be free-living algae, or they may be endosymbionts themselves living inside the cells of animals, say of corals. Corals are animals which possess photosynthetic dinoflagellates living inside them. We refer to them as zooxanthellae. This is how corals can form reefs, because they are getting most of their energy through sunlight. This would then actually be an example of a tertiary endosymbiosis, where the dinoflagellate, which has resulted from a secondary endosymbiosis, is actually living inside an animal cell. You may have noticed that secondary endosymbiosis involving brown photosynthetic symbionts have been involved in both the dinoflagellates and the heterocons, which in this family tree are closely related. Does this represent two independent endosymbioses, or might it represent simply one? Well, complicating matters is that some members of the family tree closely related to dinoflagellates, the ciliates and the apicomplexans, they do not have the ability to do photosynthesis. And so that would seem to suggest that it was two separate events. However, the apicomplexans, which include Plasmodium, which causes malaria, they do possess a plastid known as an apicoplast, which does possess genes. Unfortunately, it is hard to compare these plastids since the plastid of dinoflagellates retains only a few genes dedicated to photosynthesis, while the apicoplast possesses only a few genes dedicated to transcription and translation, so it is hard to compare them. However, there are recently discovered photosynthetic algae, which seem to be relatives of the apicomplexans. And this would tend to give support to the idea of a group known as the chrome alveolates, in which the heterocons, including the brown algae and diatoms, and the alveolates, including the dinoflagellates, ciliates, and apicomplexans, they would all represent a related group, and that a secondary endosymbiosis of a red alga occurred at the base of this group. Although the plastids descended from this lost their ability for photosynthesis in the apicomplexans, and perhaps the plastids were lost altogether in the ciliates.